my name is Madeline Arnold, and I'm a PhD student at the University of California, Berkeley. And I'm currently a fourth year in the SciJo lab, and we study the intersection between the immune system and the nervous system. And my project is looking at the role of the estrogen receptor beta in sex differences after immune activation during brain development. So just to give a bit of background, we know from human studies that if you get sick during pregnancy, this increases the chance that your child will have neurodevelopmental disorders. So clearly, activation of the immune system is somehow impacting brain development. And we can study this phenomenon using the maternal immune activation mouse model. So we take mice that are pregnant, and at a very specific time in pregnancy, we inject them with poly-IC, which is a molecule that mimics a viral infection. And then this will induce the inflammatory response in the mom and also the embryos that are developing. Then we let the embryos grow up into adulthood and measure their behavior. And what we see in this model is that the mice that receive this immune activation during pregnancy have specific behavior changes. So they have reduced social interaction, time spent interacting with other mice. They have increased marble burying, which is just a measure of repetitive behavior similar to that seen in autism in humans. But what I found interesting was that we really only see these changes in the male mice. And when you look at the female mice, which I'm showing here in orange, they don't have the same changes that the male mice do. So I'm interested in this question: what causes the sex difference in response to immune activation in this model? And my hypothesis is that the estrogen receptor could be involved in this. So the estrogen receptor is able to bind to sex steroid hormones, then go into the nucleus and bind to DNA in your cells, and can actually change the levels of gene expression. So we know that the estrogen receptor beta can actually repress the expression of inflammatory genes, reducing the inflammatory response. And my idea is that this is what could be happening in female embryos to repress inflammation and then mean that they're less likely to have these behavior changes after immune activation. So to look into this, I did a complex breeding strategy where I basically knocked out or made deficient the estrogen receptor beta in a specific cell type called myeloid cells in mice, and then measured their behavior after this maternal immune activation treatment. So what I found was that there is an interaction between sex, treatment, and genotype in these mice. It seems that the estrogen receptor beta is able to prevent the behavior changes in females after immune activation and could actually be exacerbating them in males. So I think this is really interesting. Clearly, there's a role for estrogen receptor signaling during brain development and how we respond to immune stimuli. So I'll be following up on this with the rest of my PhD. And I'd just like to acknowledge my lab, especially my undergrad, who's helped me a lot, Emily B., with these experiments. And of course, ARCS for funding me, and also the NSF. So thank you so much for listening.